Hey guys, so the first part of this unit is going to be photosynthesis, and photosynthesis is going to be broken up into two videos. This first video is going to focus on the use of light, so kind of what is photosynthesis, how does it use light, and an introduction into pigments. So the first thing that we need to understand before we get into too much detail is pretty much what photosynthesis is. And without getting into too much detail about this individual steps, photosynthesis is the process of taking in sunlight in addition to water and carbon dioxide and creating sugars. Specifically, that sugar could be sucrose or glucose or whatever the cell needs to make. And as a result of that, these plants give off a waste product known as oxygen. So that word photosynthesis literally translates to synthesis from light. So the first part of photosynthesis deals with light and how light is used in photosynthesis, and this is also known as photochemistry. So if you remember, light is a form of electromagnetic radiation, and if you look over here, you can see an electromagnetic radiation scale here. And light can exhibit two properties. It can act as a wave or a particle. And when we talk about wave, we're specifically talking about wavelength. And if you look over here in this diagram, you can see that the wavelengths are shorter up here and longer up here. And so the key takeaway is that the shorter the wavelengths, the more energy those wavelengths contain. And so visible light here is only a small part of that radiation spectrum. Also, light can behave as a particle, and the specific particle that you might remember or recall is known as a photon. And a photon is like a pocket of light energy. And this diagram here is showing what happens when a photon interacts with a molecule. So in certain cells, there are these molecules or these receptors that can receive these photons. A couple of things can happen. Either the photon could be absorbed, it can be remitted and scattered, or it can just be transmitted or pass through the molecule. We're going to focus on the absorption part. So what happens when a photon is received by a molecule and is absorbed, that molecule actually moves from a ground state to what's known as an excited state. And an excited state is also known as an unstable form. And so remember that when molecules are unstable, that means that they're more likely to react with something. So this is making molecules more reactive. The second part of photosynthesis is photobiology, and this is going to focus on the pigment part. So how are pigments involved in photosynthesis? So the first thing that you need to be familiar with is what a pigment is. And these are, pigments can absorb wavelengths in the visible spectrum. So if you look back at the electromagnetic spectrum, you'll see that visible spectrum, or that part that humans can see. And there are a couple of main types of pigments involved in photosynthesis. The biggest one is chlorophyll A, and we're going to talk about that in just a minute. We also have these things that are known as accessory pigments, or pigments that can transfer light energy to chlorophyll A. And the most common ones are chlorophyll B and carotenoids. And there are a couple of different examples just depending on what type of plant you're looking at. When we're creating graphs of pigments in the way that they can absorb wavelengths, we have two types of graphs we can make. We can make an absorption spectrum, which is measuring the absorption or the absorbance against the wavelength, or we can graph what's known as an action spectrum, which is going to be the rate of photosynthesis or biological activity against the wavelength of light. So let's focus on how pigments get their color. In this diagram here, this green line in this absorption spectrum represents chlorophyll A, the most common pigment in plants. If you can see, it is going to absorb most of the color in the blue region and the red region. So it absorbs those colors, and what's left is this green region. So this is the color that we actually see, or what is not absorbed by the pigment. So let's look at chlorophyll B, which is the blue line here. Chlorophyll B is a little bit in the blue-green area and mostly in the red area. So what we can actually see is mostly green, some yellowish color. And carotenoids are actually going to absorb most of the blue-green area, which means that they are even more yellow than the chlorophyll B is. So the key takeaway is pigments are the color that they do not absorb. Talking a little bit more about the structural differences between chlorophyll A and chlorophyll B. So they look very close to each other, and there's only a small difference here, and that's going to be that these two functional groups in these two molecules. Other than that, they're pretty much, they have the same job. Chlorophyll B is just going to trap light and then transfer that energy over to chlorophyll A. So chlorophyll A is just the more common chlorophyll that we talk about with plant pigments. And so to tie this into what we're going to do tomorrow in lab is we're actually going to separate out the pigments of a spinach leaf. And we're going to do this following a list of directions that will be provided to you. The main idea is going to be separating pigments through a process known as chromatography, which is something you might recall doing in chemistry. And depending on the type of plant that you use, we can see up to 4 to 5 bands. And each of those bands is going to represent a different type of pigment or accessory pigment. So here's an example of what a paper chromatography looks like after we run it. You're going to put a piece of filter paper and chromatography solvent, and we're going to rub a little bit of the leaf, specifically in this lab, spinach leaf, onto the chromatography paper, and we're going to watch it separate out. We might be able to see things like chlorophyll A, chlorophyll B, xanthophyll, which is another accessory pigment, or these carotene, which are carotenoids. In addition to just observing the separation of these pigments, we're going to mathematically calculate an RF factor. And RF stands for retardation factor, and this is a ratio between the distance the pigment travels and the distance the solvent traveled. And so we're going to calculate this RF value, and we'll actually be able to do a little bit of graphing with it as well, just to visualize some of the different pigments in a spinach leaf.